BioCentury This Week is brought to you by IQVIA Biotech, a full-service CRO right-sized for biotech, dedicated to helping biotech leaders close unproductive gaps in clinical development so they can protect timelines, preserve capital, and stay ahead of risk.
Welcome to a special edition of the BioCentury This Week podcast. With AACR here and ASCO on the horizon, we have the good fortune to be joined by the CMO of this month's BioCentury This Week podcast sponsor. He's Dr. James Kyle Bryan. He is the CMO of IQVIA Biotech. Kyle also serves as global head of the IQVIA Hematology Oncology Center of Excellence. Now at IQVIA, Kyle's focused on drug discovery and development in the biopharma industry with an eye on helping his clients de-risk their clinical programs. That plus his oncology focus comes in handy for us as, like some of our listeners out there, we are knee-deep in the stream of readouts from the American Association for Cancer Research meeting in San Diego. Joining me today are my BioCentury colleagues, Selena Koch, my fellow executive editor, and Lauren Martz, executive director of Biopharma Intelligence here at BioCentury. I'm Jeff Cranmer, your host, and I'd like to bring Kyle right on in and say, Kyle, welcome to the program. It's great to have you on first time.
Thank you. I appreciate the welcome, and I appreciate the opportunity to talk with you guys and talk with your listeners. I find that I am very much of a little bit of a geek when it comes to biotech and biopharma and drug development. You know, my pedigree goes back to very small biotechs that got bigger and then got smaller, worked in the CRO world for a while. I just find the whole experience is just endlessly fascinating.
Well, welcome to the Geek Club. Kyle, you are in very good company with my colleagues, Lauren and Selena. We like to get deep into the weeds on topics such as AACR. And I'd like to caution our listeners not to be too fooled by Kyle's Louisiana accent. He's actually a doctor up in the PNW, where our production engineer, Cole Travis, hangs out. Now, Kyle, I know you've got a man on the ground at AACR down in hopefully sunny SoCal, and you've been riffing with him on topics of interest. So I wonder what most immediately has caught your attention from what you're hearing.
Thank you. Yeah, we do have one of my colleagues from the Center of Excellence down in San Diego at AACR, and I'll just say he is definitely getting his steps in. He's visiting posters all over the place and tracking people down and listening to conversations. I talked with him a little bit last night and said, Jeff, what's at the top of your mind? What's your impression at the end of a long day when you're relaxing and thinking about a beer or something like that? And he was sharing with me that, both the targets and the agents that we're accustomed to seeing, the immune checkpoint inhibitors, that era of the checkpoint inhibitors is far from being over. There's tons of abstracts that are focused on both the familiar targets, pembrolizumab and the PD-1s, but also some targets that we sort of in the past had maybe underperformed in some late phase studies, but are now resurrecting either in combinations or in niche indications, things like LAG-3 and TIGIT. There's been a lot of talk about CTLA-4, kind of new generation CTLA-4 agents with maybe novel mechanisms or different ways of trying to attenuate that toxicity profile that sort of dampened what we were able to do therapeutically with those guys. Novel linkers, novel payloads, novel strategies for getting our drugs where they need to be, releasing them at the right time in the right concentration, and getting a really kind of a controlled payload release in the ADCs. Obviously, bispecific antibodies and both single target and bispecific ADCs, and then the dual payload ADCs are very big and all over the place.
Kyle, let me jump in here. We are going to dig deep into the ADC dual payloads. Lauren just did a big piece on that for us in BioCentury. But I'm intrigued about what you're saying about TIGIT. I had kind of left that by the roadside a little while ago. So I know Lauren's written a lot about this. Lauren, I'm curious, are you surprised to hear what Kyle's saying there about LAG-3, TIGIT, and the like?
Well, so I'm not surprised. And I think the thread that's underlying a lot of what you've picked out and what your colleague has picked out at AACR is antibody innovation, the bispecifics and the ADCs. So when we're talking about giving new life to some targets like TIGIT that haven't necessarily seen a lot of clinical success, especially the high profile late stage failures that we've seen for TIGIT, I think that's where antibody innovation comes into play. And specifically for that target, I think there are at least a few abstracts, and we've been seeing this ramp up over the years that are sort of dual checkpoint inhibitors, bispecific antibodies targeting maybe CTLA-4 and TIGIT. The idea there being that you could knock down two pathways that are controlling or restricting the anti-tumor immune response and, potentially making that CTLA-4 side of the equation safer. So I think that's definitely something that we've seen cropping up and something that we'll continue to see more of at meetings like this that are very focused on the innovative side of the science.
On that safety side, CTLA-4, I think, is, known for having some tox. What's promising to make a difference there? Is it masking, pH sensitivity? Either of you have thoughts on that?
I think all of the above. We're seeing some masking technologies to improve safety. And there's also FC engineering, FC tuning to try to control the ADCC and enhance the Treg depletion. So I think there are several different strategies in development to improve the safety of the CTLA-4 maps.
Speaking of safety, the other thing that is showing up are other modifications to make our ADCs safer. Novel linkers, trying to look at how to do the structure so that you get better stability of the ADC, you get a more controlled payload release. I think the tandem cleave linkers are a really interesting part there where it takes 2 steps to release the payload. So you need a cleaving that takes place at the tumor with the internalization, but this extra stabilization as the drug is circulating, it really, I think the greatest benefit there, I won't say greatest, you're letting me get too excited there, but a solid benefit there. You have to remember, I'm a hematologist and oncologist, so blood always gets me in a positive mindset, but it's reducing the hematologic toxicity because it's not having that risk of payload loss in the wrong place. The payload uncoupling is happening where it should be. And that gets you a better therapeutic index and improves your overall window.
What controls the 2 phases of the cleavage? Is there like a kind of a finite number of different ways that's accomplished?
I hesitate to say finite just because I feel like, the only thing we're limited by is our imagination. But some of the same techniques that are being used in masking, where they take advantage of the fact that, there are differing enzyme levels at the tumor microenvironment compared to what you're experiencing in the circulation, and utilizing that as that initial cleave taking place when you're entering in the tumor microenvironment. And then whether it's the target or the payload being freed up, then to go through that second cleaving after it gets internalized.
I mean, ADCs are so interesting in their complexity, the modularity of it. You can do all kinds of antibody engineering, as Lauren was talking about, on the antibody end, multiple specificities, whatever. Then there's the linker, which sounds like those are getting more complex as well. And then there's the payload. There's like any number of cytotoxins, but there's radioisotopes, on and on. Now there's also this dual payload approach, right? Maybe we should talk a little bit about the increase in abstracts this year and what we're seeing for dual payloads at AACR.
Yeah, that's a good call there. Lauren just wrote about that. Lauren, why don't you jump in here?
Sure. Thanks, Jeff. So the dual payload ADCs is one thing that we've picked out as a trend in the abstracts from this year. There were, I think, more than 40 abstracts that discussed this idea, which is something that we've heard a bit about, but again, really taking off in this crop of abstracts. These were broken down by, or we broke these down by dual payload ADCs that are delivering 2 cytotoxic chemotherapy payloads and some that are delivering, one of the standard cytotoxics plus a targeted therapy or something else like a degrader. I think both of those were really interesting approaches because the 2 cytotoxic chemotherapies, a lot of times it's combining the standard therapies that ADCs deliver. And the idea is that you could overcome resistance that ultimately develops with most of the ADC treatment. Even though these are really potent therapies, it's still a chemotherapy and tumors can still sort of evolve resistance to get around them. And then I thought the chemo targeted therapy combinations were really interesting because it just offers one more way to, first of all, to combine 2 things and avoid resistance, but also to localize the effects of the targeted therapy. In the news last week, we saw the Revolution Medicine data for their pan-RAS inhibitor. That was just phenomenal data, but we do know that there are some side effects. So, one of the abstracts that we saw in this crop this year from Affinity Biopharma delivers a pan-RAS inhibitor. And you think, well, maybe this is a way to sort of better localize that pan-RAS effect and avoid some of the side effects. We don't know yet. This is all preclinical, but it's just a really interesting crop of abstracts.
Yeah, those are really good points. I mean, one of the things I think that's really interesting about the dual payload ADCs is sort of the steps that you have to take through in the development as you look at this, because again, the concept is really strong. I love the concept behind, for example, if you're using a DNA damaging agent along with a DNA repair inhibitor, and that gets to the whole idea that the reason the ADC fails or the treatment fails is payload resistance. It isn't actually loss of target, which all of us that have worked in the antibody world are always, what happens to the expression of my target over time? Is that changing? Is this why my agent isn't becoming as effective? But when you're looking at 2 payloads, you've got to think about the individual toxicities of the agents and the risk of overlapping toxicity of the agents. But then the extra wrinkle is, well, what if there's a new synergistic toxicity that I don't see with A and I don't see with B, but when I put A and B together, I now get C, which I wasn't expecting. So as I work through, the dose escalation and the dose finding and dose optimization aspect of it, it just becomes one of those things where we need to keep our eyes wide open, looking for those safety signals.
That brings up another point that I heard in the reporting for this story, which is, yes, the combinations, we have to assess the safety, but even once you figure out the right combinations and the right doses, actually creating an ADC that is able to deliver the right ratio of these payloads is really challenging, depending on which type of linker you use, whether both payloads are on the same linker, it's a heavy lift and a really interesting challenge.
Yeah.
Do more of these abstracts that you're seeing at AACR have branched linkers, or do they connecting them up separately?
That's not disclosed in all.
It's often not disclosed.
Yeah.
I hear that a lot of them are separate linkers, but I can't confirm that.
Yeah, there's not a lot that they've talked about yet about, for example, the DAR on these agents and, what's the DAR of this payload and what's the DAR of that one. It is one impacting the other. So there's a lot for us still to learn. And again, the engineers are kept very busy.
I think the segue is really nice into a topic that I know is on your mind lately, de-risking. So you have, like I was saying, this complex modular modality where you could tweak any piece of it in lots of different ways. The human imagination can do almost anything there, right? The combinations are staggering, the possibilities. But how do you think about smart de-risking?
The way I tend to think about it is like this. I think when it comes to drug development, and particularly my pedigree is in the biotech world and sometimes in the very small biotech world, where when it comes to evaluating an agent or an antibody or a compound or any investigational product, you have to be a little bit careful of being more paternal than professional in your evaluation of it. You do get this feeling it's your baby, but just recognizing that, it may be a smarter thing to think of it like when you're making breakfast. You know how the first pancake never turns out all that great? and you may need to toss that one away and focus on the rest of the batch. That's kind of the thing. So I would say that one of the bits of advice that we give our biotech clients is, again, look at it professionally. Consider like a stage gate analysis where you're really looking at every step in this development and setting parameters for what you're going to do if you see this or what you're going to do if you see that, as opposed to, well, let me see what happens and then I'll think about what my decisions want to be. Because you've got to be looking at, is the science credible? So what's the science and the tech readiness of this is one point. Is the risk acceptable? Company-wide, the risk to a patient, the risk benefit aspect of it, and to do a real risk assessment that you're then going to apply to your decision-making at each stage as you go along here. What's the investment? Is the value there? Do the economics work for me? And then finally, you can't forget, is this a strategic fit for my company, for my group that are developing it? You may see something really interesting that can carry an exciting new direction. But if the strengths of your team aren't in that kind of development, then you're either going to need to bring in new team members, or you're going to need to think about, well, maybe this is something that I hand over to someone else to develop at this point, or I look for a different kind of partner, a different kind of engagement to take it to the next phase. I think that too often, especially in our smaller biotechs, that you can get in a more of a state that where your overview of the program, it looks more like a project review or an operational review where you're tracking progress as opposed to having a real decision-driving mindset. And these strategic discussions of shall we continue or not continue, as opposed to making the little tweaks around the edge. So that, I guess, would be my broad approach to how you de-risk and think about this. Ultimately, we want people to focus on trying to eliminate the white space. A lot of the unproductive part of drug development comes from not the time to get to a milestone, but getting to a milestone and then the delay in coming to a decision or deciding what you're going to do with the information that you receive. And I think that, again, it's a place where it's important to look at your team, look at your advisory persons, whether it's your board or your scientific advisory board, and kind of build in that sort of upfront planning, smart decisions about your protocol to keep it in scope with what you can accomplish, and really use the data and kind of cross-functional alignment to get to a good decision point.
All right, some good practical advice there from Kyle. We're going to take a quick break, and then we're going to come back and look ahead to ASCO.
BioCentury this week is brought to you by the 26th Bioequity Europe in Prague, Czech Republic. Innovators everywhere are seeing speed as the driver of investment, partnering and success, speed to the clinic, speed to early human data, speed to de-risk assets. The question for European biotechs is clear. How do you compete in this arena? Facing new momentum from Asia and an unpredictable landscape in the US, the science in Europe is there. There are more experienced operators. And investor sentiment finally is turning. What's needed now is speed. In its 26th edition, Bioequity Europe travels to Central and Eastern Europe for the first time to explore emerging opportunities in the region and debate how European biotech can accelerate to compete and lead in today's world. Join us in Prague, May 4th to 6th for this destination event for investors and biopharma leaders. Register at bioequityeurope.com. Bioequity Europe, where Europe accelerates to compete.
Okay, we are back on the BioCentury This Week podcast. It's a special edition, and we are joined by Dr. James Kyle Bryan, who is the CMO of our sponsor, IQVIA Biotech. We're going to get Kyle's thoughts in a moment on how he's getting ready or thinking about ASCO, which is, it's next month, so it's not too far away. AACR just runs into ASCO, though you do get that little digestive disease week aperitif in between. Lauren, before we get there, do you want to just give us a few takeaways just wrapping up the ADC thoughts?
Sure. Yeah. Just to wrap up what we talked about, we've gone through the dual payload ADCs as actually the bispecific dual payload ADCs are sort of the tip of innovation right now at AACR. We found 44 abstracts that cover dual payload ADCs. A lot of them are bispecifics. And just looking more broadly, because Kyle has a focus on antibody innovation in his past, there's a lot more in those abstracts. There are tri-specific ADCs and masked biologics and all kinds of really interesting ways to innovate and take different approaches to risk, as Kyle has talked about. With these dual payload ADCs, it's a lot of the same targets that we've seen for the approved ADCs when you're talking about the tumor antigen. It's a lot of the same payloads, but just combinations of payloads. So you can see companies and institutions, although most of these are from companies, thinking about where they're going to apply that risk, which
Lauren,
is really the technology.
Is anyone going out on a limb there and kind of stacking up a little bit of risk, say putting a new target on with a dual payload? Or
That's a great question. There are quite a few new targets, and by new, not new to us as people who are looking at the innovative edge of everything, but new as in no ADCs approved against this target. Certainly a lot of new tumor antigens that are in the list, really a lot of the same cytotoxic payloads that we've seen, but there is some innovation. For example, a company called Baylink has a dual payload ADC targeting GPC3, and one of the payloads is a protein degrader. So lots of risk in that one, but huge potential reward, I guess, if it works well.
And you can check out Lauren's article on biocentury.com. I'll drop a link in the show notes. I'm going to bring Kyle back in here. Kyle, how are you thinking about ASCO from where you sit at this time of year?
Well, you can't really be at AACR without thinking about ASCO. In my mind, the way I think about it is that AACR has always been about the strategic direction of cancer research. Where are we going strategically? ASCO has traditionally been about clinical practice. What are we doing directly for patients? Is this going to change clinical practice? Not that we don't see preclinical data at ASCO, because we do, but that's sort of what I call the pull through, the AACR to ASCO pull through, and what likely to happen. So I really think for this year's ASCO, we're going to see more about the dual payload ADCs, some of the early clinical proofs, or at least early discussion on this. I think, again, this narrative of where the ADC resistance originates, that narrative, I think, is going to be picked up, and we may see some additional interesting discussion around that. Ultimately, we need to see the phase 1 expansion data, and it's possible we'll have some of that. More likely, we're talking about pull-through for 2027 ASCO, but it's just a really interesting period there. I think looking at other combinations, whether it's with checkpoint inhibitors or whether it's with chemotherapy, I think those are going to be there. There's a couple of things that we didn't really even touch on today that will probably be featured when we're looking at the data at ASCO. We didn't even get a chance to talk about the mRNA cancer vaccines, or talking about maybe the AI-derived biomarkers and what's happening in that space. We'll have an exciting ASCO. The amount of data there is way more than you can consume in 4 or 5 days. So that's why we all end up reviewing the abstracts and watching the virtual meeting for months to come. But it's really been an exciting AACR so far, and it's not over yet.
Excellent, Kyle. Well, thank you for sharing your thoughts. Would love to reconnect with you after ASCO just to see what the big takeaways were for you there. And always grateful to IQVIA Biotech for sponsoring the BioCentury This Week podcast. Kyle and Lauren and Selena, thanks so much for sharing your thoughts on AACR. And Kyle, looking ahead for us to ASCO.
It was wonderful, Jeff. I really enjoyed talking with you and with Selena and Lauren. I'll look forward to connecting with you again soon.
Okay, you've been listening to the BioCentury This Week podcast, special edition here with Dr. James Kyle Bryan, CMO of IQVIA Biotech.
BioCentury would like to thank IQVIA Biotech for supporting the BioCentury This Week podcast. To learn more about how IQVIA Biotech's dedicated teams deliver support from innovation to impact, visit IQVIAbiotech.com.